Hello and welcome to part 2 of chapter 8. We last left off finishing section 8.4 talking about a photosystem. Here is a different image of a photosystem from the one I showed you in the first video, but basically it's the same thing. And this is the beginning or most of the light-dependent reactions. The light-dependent reactions occur in 4 stages. Stage 1 is the primary photo event. This is when photons of light are captured by the pigments. So here you see the light coming down and being captured by the pigments in the antenna complex. Stage 2 is called charge separation. Energy transferred to the reaction center, which sends an electron to an acceptor molecule. So the energy that builds up in the reaction center, once it builds up to a critical level, it will eject the electron up and it gets accepted by a primary electron acceptor. Stage 3 is electron transport. The electron is then passed through an electron transport chain to generate a proton gradient. The final electron acceptor in this electron transport chain is NADPH. Stage 4 is chemiosmosis. That proton gradient is used to generate ATP using ATP synthase, the same way it was done in aerobic respiration. This process can also be called photophosphorylation. Phosphorylation, we already learned, is simply taking a phosphate and sticking it on ADP to make ATP. In this case, the energy used to do that is light energy, hence the word photo in photophosphorylation. So there are 2 types of photophosphorylation. The first type is cyclic and the second type is non-cyclic. So let's look at cyclic photophosphorylation. This involves one photosystem to generate ATP. Organisms like purple non-sulfur bacteria and green sulfur bacteria do this type. They eject an electron. As it's passed through an electron transport system, the proton gradient drives chemiosmosis, just like I described previously. But in this case, that electron, instead of going into NADPH, it's recycled back into the reaction center. So no NADPH is formed and no oxygen is formed. So this method is anoxygenic. So in this diagram, here's our photosystem 1. It absorbs the light, photons of light coming from the sun. The electron is ejected out of the photosystem into an electron acceptor, which then passes it along an electron transport chain, and it goes back in to the spot where it held before. Instead of having this space filled by an electron donor, and instead of it being accepted by NADPH. The second type is non-cyclic photophosphorylation. And let's take a look at how that looks. Okay, so this system uses 2 photosystems. Now, some students get confused by the fact that it has photosystem 2 listed here and then photosystem 1 listed here. They were numbered based on when they were discovered, not where they're located. But don't let that confuse you. All of this happens at the same time, not in a stepwise fashion. So light strikes both photosystems at the same time. Excited electrons are ejected out of the photosystems, and in this case that leaves a hole. The electron needs to be replaced. So the electron that's ejected out of photosystem 2 passes down an electron transport chain and gets accepted by photosystem 1 to replace the one it lost. The electron that photosystem 1 lost also gets moved down an electron transport chain, but it gets accepted by NADP to become NADPH. This still leaves an electron hole in photosystem 2. It needs to replace its electron that it lost. That's done by splitting water. So water is broken apart, electron is donated to the photosystem, and that's what produces oxygen. That's why the cyclic photophosphorylation does not produce oxygen and the non-cyclic photophosphorylation does. So now let's try to get a complete picture of this on the thylakoid membrane. Now I'm afraid this image kind of got distorted and I've been unable to undistort it, so we'll have to deal with it as is. Okay, so let's put the story together. Here's photosystem 2, here's photosystem 1. Photons or light are striking those photosystems. They are ejecting electrons. The electron coming out of here is going through this electron transport chain, which is pumping hydrogen ions into this thylakoid space, and it's replacing the electron lost in photosystem 1. The electron from photosystem 1 is going out through here and being accepted by NAD to generate NADPH. The electron lost from photosystem 2 is replaced by water, and that's what's generating our oxygen. Hydrogen ions then build up in this thylakoid space, creating a concentration gradient between that and the stroma outside. Hydrogen ions will then diffuse through the ATP synthase complex, generating ATP. And you now have ATP and NADPH being made. Those are the two products of the light-dependent reactions, along with the byproduct of oxygen. Oxygen gets released by the plant to our benefit, and the ATP and NADPH are now holding energy that can be used to run the light-independent reactions. So the light-independent reactions actually take that energy and grab carbon dioxide gas and take that carbon and convert it into an organic molecule. So let's look at that. So this is called the carbon cycle. It happens in C3 photosynthesis. It occurs in the stroma of a chloroplast and uses ATP and NADPH to reduce carbon dioxide into organic molecules. Now, I know this diagram can look very intimidating with all these long, complex words, but you don't need to know all of that. We're going to break it down so that you understand what you need to know. The Calvin cycle has 3 phases. Phase 1 is carbon fixation, the incorporation of carbon dioxide into organic molecules. It begins with a molecule called RUBP. You don't need to know the full name, that's it written here, but 6 RUBP molecules get combined with 6 CO2 molecules here, and this produces 12 molecules of PGA. Okay, you don't need to know the full words, just the initials are fine. This is phase 1. Carbon fixation, and it uses an enzyme called Rubisco. If you can't read that, I'll write it up here. Rubisco. I always think of Crisco for some reason when I hear that word. Phase 2 is called the reduction phase. It takes the 12 GPA molecules and reduces them into 12 molecules of G3P. This process uses ATP and NADPH. You can see that here. Hopefully you'll recognize the G3P. It was an intermediate or substrate in the glycolysis pathway. So it is an organic sugar. 2 of these G3P molecules will actually leave the Calvin cycle and be converted into glucose or other sugars. Phase 3 is the regeneration of RUBP. Those 10 G3P molecules are used to regenerate the 6 RUBP that can then go back and collect 6 more carbon dioxide molecules and keep this cycle going. And this process also uses ATP. So that's the Calvin cycle in a nutshell. It just continues to turn, pumping out G3P molecules that are used to make sugars. Please note that glucose is not a direct product of the Calvin cycle. It's actually G3P that's moved into the cytoplasm where they are then used to produce sugars. Energy for the Calvin cycle is supplied by 18 ATP molecules and 12 NADPH molecules, which were produced in the light-dependent reactions. So that's photosynthesis, converting light energy into organic molecules, trapping the energy in an organic form. So now let's look at the big picture. Essentially, mitochondria and chloroplasts are dependent on each other. Chloroplasts produce glucose and oxygen, and those are the starting substrates of aerobic respiration. Glucose and oxygen are broken down to generate ATP. The byproducts of this are water and carbon dioxide, which are the starting products for photosynthesis. The water is used to donate an electron to a photosystem, and the carbon dioxide is fixed into glucose. You can look at this as one big system of transferring energy. Sunlight energy is trapped by the chloroplasts into an organic molecule, and when that's needed for use to do work, the organic molecule is broken down in respiration to generate ATP, which actually is the form of energy that's used to do work in a cell. Okay, so this takes us through section 8.6 of photosynthesis. I'm going to do one final video to cover the remainder of the chapter. Thanks for watching.